conflict of interest. Each of you will have 5 minutes. And actually, this is the part of sharing that I don't like to do, but we will try to stop at the 5 minutes. So please be conscious of the time. First is Kina Gooden, Director of Worldwide Health Economics and Outcome Research in BMS. Dr. Gooden. Can you hear us?
Dr. Gooden, we see you, but we can't hear you.
Is this better?
Yes, we can hear you now.
Great. Thank you. So again, my name is Kina Gooden, Executive Director for Worldwide Health Economics and Outcomes Research for Hematology and CAR-T at Bristol Myers Squibb. I have no additional conflicts of interest. Thank you for the opportunity to speak to the value and importance of idacabtagene vicleucel, or ide-cel, a CAR-T cell therapy approved by the FDA as a BECMA. Abecma is approved for the treatment of adult patients with relapsed or refractory multiple myeloma after 4 or more prior lines of therapy. Abecma is a breakthrough CAR T cell therapy that offers triple-class exposed patients who have limited effective treatment options a potentially transformative personalized therapy. The way BMS assesses the value of its medicines, including Abecma, is fundamentally different from ICER's. Even within this specified criteria of this review, there are several concerns. Namely, there are challenges associated with reviews that are announced in the early stages of a medicine's life cycle, given the fast evolving body of scientific evidence. In this case, ICER's timelines did not allow for a full and thoughtful consideration of publicly available data in 2 key areas. 1, ICER underestimated overall survival, and 2, ICER overestimated the impact of retreatment. The first point, ICER underestimated overall survival. Overall survival is the clinical gold standard used by many health technology assessments for measuring a patient's response to a medicine. The underestimation of overall survival led to lower qualities attributed to Abecma and an overall underestimation of its value. Specifically, ICER inappropriately used progression-free survival to estimate overall survival and ignored existing evidence in the public domain, that evidence being longer-term survival from the phase 1 CRB 401 study. The overall survival for CRB 401 is 34.2 months. Abecma's impact on overall survival is supported by data in the KARMA trial, published in the New England Journal of Medicine, which showed 12-month overall survival was 78% of 128 patients, and a large number of these patients continue to exhibit a complete response. Our second point, ICER overestimated the cost of retreatment based on a misguided interpretation of data from the KARMA study. While some patients were retreated with Abecma, responses were infrequent and less durable when compared to initial treatment. Based on this limited clinical trial experience, it is not appropriate to assume retreatment in the real-world setting. If you look at the scenario analysis, you'll see that patients who were retreated were assigned full costs associated with CAR-T in the ICER model, such as infusion and other resource utilization. Since there is limited data, this assumption is not accurate and is a driver of ICER's model results. We believe these concerns are valid and rooted in sound clinical, scientific, and economical expertise. Had ICER properly accounted for these nuances, we believe it would affect its cost-effective findings. It is important to note that mature data has yet to be fully made available to the public, and thus reviews like this risk being premature altogether. Long-term follow-up of the pivotal trial is expected to be reported in the following months. We respectfully urge ICER to consider the complications that occur with reviews announced in advance of FDA approval, given the modeling and methodology issues outlined. We also encourage ICER to make their modeling and methodology frameworks transparent and their use of publicly available data consistent across reviews. In standard practice with our approved medicines, we look forward to making our cost effectiveness analysis transparent in the near future. We remain confident in the value of Abecma and the hope it brings to patients and the future of multiple myeloma treatments. Thank you.
Thank you, Dr. Gooden, and thanks for staying on time. I want to ask if anyone from ICER staff would like to respond to that comment. And maybe specifically, Dan, do you have anything you would like to say?
Yeah, thanks, Reem. And I should introduce myself because I haven't had that opportunity yet. I'm Dan Ollendorf, Assistant Professor at the Tufts University School of Medicine. I'm also working as a project consultant to ICER on this review. And so we appreciate the comments from Dr. Gooden, and we also have appreciated the engagement with BMS throughout this project. The first response around the immaturity of the data and the speed of this review. This comment does come into ICER on a fairly frequent basis, but we have to continually remind folks that the rationale for doing the reviews on this timeline relates to the fact that decision makers, particularly clinicians, patients, and payers for that matter, will be making decisions based on the same set of data that we're looking at. So we do not think this is an immature or process that's too early because of that. In relation to the survival data, again, we've appreciated the conversations we've had with BMS. Some of those conversations that led us to modify the modeling approach because of the underestimation of PFS that came out in the draft report. As Brett noted in his comment, we are now actually overestimating PFS to a certain extent related to the KARMA trial. We did receive input about potentially using the phase 1 CRB 401 study, and we decided not to pursue that for focus on the clinical effectiveness review or the modeling, because in fact, there was a percentage of patients who are not actually triple class refractory in the CRB 401 population. And so the patient group that's of focus in phase 1 in the phase 1 study is really not the patient group that we're focused on for this particular review. And so we felt that the KARMA trial data, despite its limitations, better reflected the population that we're focused on. In terms of retreatment, we note that again, despite the fact that we had multiple conversations and there were conference presentations on the KARMA study, retreatment was never even raised as an issue until the publication of the trial in the New England Journal, which came out between the publication of the draft report and the revised report. We asked for additional data to inform our final evidence report on retreatment, did not receive any. And so we were limited to the 1 paragraph description in the New England Journal about retreatment. So we did make an assumption about a second charge for retreatment. As Brett noted, we did a scenario analysis where that assumption was removed. And while results did change, we noted that the cost effectiveness numbers did not reach common benchmarks for cost effectiveness. So we tried to be as flexible as possible with that, with a lot of uncertainty about how retreatment would be considered in practice.
Reem, can I just, the last issue is around transparency of the modeling. If you go to the supplement, we list all of our survival distributions that we used for the base case. We provide all of the survival extrapolations against the observed data. We're not hiding anything. It's all there. Anyone really that knows modeling can replicate it. So I don't think there's any issue with transparency in this report.
Thanks, Britt. So with that, I would like to invite
Can I address any of the comments or no?
I really apologize. We have to make sure we stay on time. We're already behind.
Okay, thank you.
Thank you. Dr. Era Gupta, who's the Vice President and Medicine Development Leader of GSK. And please go ahead. You have 5 minutes.
Okay, thank you for this opportunity. My name is Ira Gupta, and I'm the Vice President and Medicines Development Lead for Global Oncology at Glaxosmithkline. Thank you again for the opportunity to give feedback and participate today, as well as over the course of the assessment. I do not have any further conflict of interest to disclose. Relapsed and refractory multiple myeloma is both incurable and devastating for those living with the disease and their loved ones. There is significant unmet medical need in this late-line indication, where patients often run out of possible effective treatments and have very poor survival outcomes. According to the ICER's comparator in this report, the median overall survival is only 7.7 months. Most treatments are combination therapies, such as triplets, with limited data in late lines of multiple myeloma. Patients suffering from this incurable disease therefore need additional options. Blenrep, or belantamab mafodotin, is the first anti-BCMA therapy that is approved in multiple myeloma. It is an off-the-shelf, single-agent therapy with a novel mechanism of action. In the pivotal DREAM2 trial, patients who were administered the approved dose of 2.5 milligrams per kilogram every 3 weeks had a median overall survival of 13.7 months, which is approximately 6 months longer than the survival of ICER's standard of care comparator. The DREAM2 population had a median age of 65 years and had received a prior of 7 prior lines of therapy. In this late line population, with an expected overall survival of only 7.7 months, a key parameter to consider is the duration of response. Of the 1/3 of patients that responded, patients experienced a clinically meaningful duration of response with a median of 11 months, which then translates to the improvement in overall survival. One of the key aspects that I would like to focus on are the corneal events, which have been reputed in the DREAM2 study. The point I want to bring up is the fact that these eye examination findings or keratopathy do not always lead to a patient-reported adverse event. No permanent complete vision loss was reported, and only 3 patients discontinued due to the corneal events. 18% of the patients experienced clinically meaningful changes in best corrected visual acuity, that is defined as a loss in visual acuity to worse than 20/40 in the better seeing eye. These events resolved with dose holds and reductions and are not likely to be associated with a high cost of management, as no patients experience hospitalization due to the ocular events. We also note that the overall 10% discontinuation due to adverse events, which was reported in the DREAM2 study, is lower than the 14 to 18% rate, which has been cited by the 3 usual care regimes in table 3.7 of the ICER's report. We also believe that the ICER's comparison of Blenrep median PFS and median OS that is reported in the DREAM2 to a fixed ratio is not conventional when overall survival data is available from a clinical trial. Felix 2013 is the full peerly reviewed publication that ICER has cited. And in that report, the average ratio of median time to progression to overall survival was 5.3 times in a relapsed refractory patient population in phase 3 studies, with a range of 2.8 to 6.9 times. The ratio for DREAM2 trial is therefore consistent with the data in this comparable trial patient population. In terms of the patient's quality of life, ICER notes Blenrep treatment was associated with stable health-related quality of life, with trends towards improvement in some domains. Patients entering the DREAM2 trial had a low baseline quality of life score, which is lower than in all other multiple myeloma trials. The appropriate expectation versus baseline in such a heavily pretreated population should be one of maintenance of quality of life, and treatment with Blenrep meets or exceeds this expectation. As an FDA-approved regimen, Blenrep has undergone detailed benefit risk assessment, and the FDA Oncology Drug Advisory Committee, which includes oncologists, ophthalmologists, patient advocacy groups, etc., voted 12-0 in favor of a positive benefit risk. This was based on deep and durable responses with the monotherapy that led to significant improved survival compared to the standard of care and manageable safety profile. To conclude, Blenrep offers patients with heavily pretreated multiple myeloma a much needed and innovative treatment option. Thank you again for your time and the opportunity to comment on behalf of GSK.
Thank you, Dr. Gupta. And again, thanks for staying within your time. C or Brett, anyone would like to comment?
I'll take a crack at some of the points that were raised. So I guess the first point that I heard was kind of how favorable the overall survival results were for belantamab, and certainly I would absolutely agree with that the overall survival results was the most favorable finding in the DREAM2 study. But as I alluded to earlier, it feels to me that the most objective way to evaluate the response is really looking at not just overall survival, but things like progression-free survival, as well as overall response rate. And some of those other metrics, I don't think belantamab looks as good. So I think it's, I agree that the overall survival results are favorable, but I think it should be reviewed in context with some of the other results. Regarding the corneal events, I think this is one of those situations where it's, we agree that the corneal events, that the overwhelming majority of patients, for them, it is reversible. But as I noted earlier, when you're talking about patients with a limited remaining life expectancy, having a duration of visual deficits for a month of that, I think is a significant quality of life hit. Regarding the point about that the metric that we should be using for health-related quality of life is not declining, I think that's something that is an interesting position. It's not something that is, I don't think it's standard to view quality of life in that way. And I would certainly love to see what the health-related quality of life measures are for usual care, and I think that's really the comparison that we should have. But I do think that, and this is why, given the input from GSK, we inserted that language in the report about it being stable. But I do think that you know, I would say that I agree with that point, that it is stable. Whether just maintenance is good enough, I think that is open to question, and that's something that the panel will have to decide. I'm going
to ask you for maybe one last point because we have to move on.
I will stop and see if Brett has anything that he wants to add.
Just the only quick note is that we did pick the base case as observed from the trial, and that's why the scenario was used and not the other way around.
Thank you. Now we're going to go to public comments. David Hendricks, again, please stay within the 5 minutes. We are behind schedule for about 5 or 6 minutes, so we'll try to wrap up for sure by noon Central.
Okay, so you can hear me?
Yes.
Okay, well, I'm David Hendricks. I'm a patient. I'm surprised I'm the only patient speaking here, but no matter. So I thought I'd just speak of my experience with myeloma and then my experience with the CARTITUDE trial and how life has been since then. So I was diagnosed with MGUS in 2010, which rapidly progressed to myeloma, and I was diagnosed in 2011 with myeloma. My doctor is Dr. Tom Martin at UCSF. Started first induction therapy in October 2011, classic Revlimid, Velcade Dex. It worked very well. Knocked me into remission, and then I went on Revlimid maintenance. I was on maintenance for a couple of years. At one point, I was down to complete remission, 0 spike, and all the numbers were low. At the end of 2015, Dr. Martin took me off Revlimid. I thought I went in a year without Revlimid, but in 2016, it started coming back. And since then, it's been a succession of different therapies that have all worked for a while, then didn't work. Or I should say some of them didn't work so well. Second therapy was in the spring of 2017, ixazomib, Revlimid, Dex. I just did 5 cycles of that, wasn't really working. Then in the fall of 2017, went on daratumumab, pomalidomide, Dex. And that actually was a fairly good experience. I'd say the side effects were less severe than the other regimes I'd been on before. And I just, in the end, was going into UCSF every 2 weeks and then every 4 weeks for the infusion. It was really nice, but unfortunately it didn't work. And apparently my myeloma doesn't respond to drugs that target CD38. And unfortunately, my numbers got pretty high. And in spring of 2018, I had compression fractures in my vertebrae. This is the first time I really had physical symptoms from the myeloma. So that was a pretty painful spring. I had 2 kyphoplasties to repair that. In the summer of 2018, I started on carfilzomib, cytotoxin, Dex. And I didn't think it was going to work because carfilzomib is in the same family as Velcade and ixazomib, but actually it did work. Knocked my numbers back down again briefly, but then they started climbing again. So in 2019, my numbers were climbing again. This is my 4th therapy. I was turning out to be refractory to everything tried. And at this point, Dr. Martin proposed that I participate in the CARTITUDE-1 trial. So I had the infusion on November 20th, 2019 at UCSF. And I would say, the infusion itself was kind of a non-event. It was just 30 minutes in and done. And for a couple of days, it was like staying in the hotel, no problems. But then on the 5th day, I started spiking a fever, and then I went through a pretty painful 4 days, cytoxin release syndrome, CRS. My fever spiked up to 39.5. They gave me tocilizumab once. And after my fever came down, my ALT and AST spiked. So they were concerned about my liver. So I needed to stay in the hospital longer till that came down. So eventually I was out. And I would say since then, I had a bone marrow biopsy in January of this year, which is over a year after the infusion. And they could not detect any plasma cells in my bone marrow. And that means no normal or abnormal. And Dr. Martin said he'd never seen a case of no normal plasma cells. At the time, my IgEs were all very low. Since then, my IgEs have started to climb again, which is good news. It means hopefully I'll have a reaction to the COVID vaccine. Side effects from the CAR T, I had swelling in my ankles and feet for a couple months afterwards, but that eventually went away. And the other major symptom is loss of sense of smell. And Dr. Martin has told me, apparently this is not a widespread side effect, but it's happened a lot to the patients who had the infusion at UCSF. That doesn't bother me because I think the benefits greatly outweigh the kind of the annoyance of losing my sense of smell. I mean, when you're on chemotherapy for long periods of time, you don't realize how unpleasant it is. It really messes up your sleep, messes up your digestion. And it kind of goes on and on, makes life kind of tedious at times. Since over a year now, since I've had the CAR T, no chemotherapy, I can eat almost anything I want. I don't get sick anymore. I don't have the frequent constipation that I had before. I sleep better, and quality of life is much better. And I'm not going into the clinic for infusions and taking expensive drugs. So for me, it's been a great experience.
David, thank you very much for being here and for your willingness to share this story with us, which gives us the perspective from patients. Clearly, this medication has changed your life, is what we're hearing from you.
Mm-hmm.
Thank
Yes.
you very much. With that, I think this is time for break now. We are going to come back at 12:30 sharp, so please be here a couple minutes before just to make sure we're on time then. Anything else, Steve?
No, that's perfect. Thank you, Reem.